The next part of this lecture is looking at coding for stimulus intensity, and all action potentials are going to be alike in that they are showing a change of 100 millivolts, so they're independent of the original stimulus strength. Them being an all or nothing phenomenon means that they stay the same strength throughout the entire axon as the signal moves from the axon hillock to the axon terminals. So strong stimuli can generate nerve impulses more often in a given time interval than weak stimuli. That means if you have a stronger gradient potential, we will get multiple action potentials, but we won't see an increase in the strength of that action potential. So we code for stimulus intensity by increasing the number or frequency of action potentials, not their strength. So strong stimuli cause action potentials to occur more frequently. Stimulus intensity is coded for by the number of impulses per second or by the frequency of action potentials. And so we're not looking at the strength again for action potentials, we're looking at how many occur over a certain period of time. Here we can see in figure 11.13 that stimulus 1 doesn't reach threshold, so there are no action potentials. Stimulus 2 reaches threshold, but is weaker than stimulus 3, so there are less frequent action potentials for stimulus 2, which is weaker than stimulus 3, and stimulus 3 has many action potentials that occur over the same amount of time. So if we were measuring the number of action potentials and trying to figure out how strong was the original stimulus, we would say that stimulus 2 must be weaker than stimulus 3 because there were less action potentials during or because of stimulus 2 than because of stimulus 3. Absolute refractory periods are the period of time from the opening of the sodium voltage-gated channels until the start of the closing of the potassium voltage-gated channels. So we would see that during this period of time that another stimulus or another action potential cannot occur. The absolute refractory period occurs at the same time as the action potential being generated. So this is why we see action potentials unable to increase strength. They're always going to be represented as a change of 100 millivolts because they cannot occur on top of each other. So we're preventing the neuron from generating more than one action potential at one time because of this absolute refractory period that prevents more than one action potential during the same period of time. This ensures that action potentials are separate from one another, and that's when we measure their frequency to look at stimulus strength, then how tall or their difference in magnitude. It also enforces one-way transmission of nerve impulses from the axon hillock to the axon terminals. There is also a relative refractory period, which is a period of time when usually another action potential would not occur. It's when the voltage-gated sodium channels are still closed, but the potassium voltage-gated channels are open. We're usually in the hyperpolarization stage during this time, but during the relative refractory period, if we had a very strong stimulus, we could have another action potential during this time. But unless the stimulus is very strong, another action potential would not occur during the relative refractory period. But unlike the absolute refractory period, an action potential could occur, is just unlikely. So here we can see the refractory periods. We see that the absolute refractory period occurs at the same time as the action potential. So this ensures that we don't have two action potentials at once. They don't build on each other in strength, and therefore an action potential always a change of 100 millivolts. And then the relative refractory period occurs during the hyperpolarization stage. And this is when an action potential can occur, but is unlikely to unless the stimulus is strong enough. Earlier in this PowerPoint, we talked about conduction velocity. We know that the axon diameter and the degree of myelination greatly increased conduction velocity as long as the axon was large and it had myelin. Continuous conduction is slower because it occurs in non-myelinated axons, whereas saltatory conduction is faster because it occurs in myelinated axons. We also saw that larger diameter fibers or axons had less resistance to current flow, so we had faster impulse conduction in large diameter axons. So just remember that conduction velocities of neurons vary, and if we take those two characteristics into account, axon diameter and degree of myelination, we can create nerve fiber classification. The nerve fiber classifications take into account the diameter of the axon, the degree of myelination, and then as a result of those two components, the speed of conduction down the axon. So we have group A fibers, group B fibers, and group C fibers. Group A fibers are myelinated and have the largest diameter of these three classification groups, so they also have the fastest speed of conduction at 150 meters per second. Group B fibers are only lightly myelinated and they have an intermediate diameter, so we're seeing a decrease in myelination and axon diameter. As a result, they transmit signals slower at 15 meters per second, whereas our group C fibers have the smallest diameter and no myelin. So they transmit speed of about 1 meter per second, so their speed of conduction is much slower compared to group A and B fibers. Another topic we want to acknowledge are synapses. Synapses occur between neurons or neurons and effector cells. In Chapter 9, we saw a synapse occur between a multipolar motor neuron of the somatic nervous system and the sarcolemma of a skeletal muscle cell. So presynaptic, there would have been a motor neuron. Postsynaptic, there would have been a skeletal muscle cell. If we're talking about two neurons, the presynaptic neuron is the first one that conducts impulses towards the synapse or the space. You may remember the synapse being called the synaptic cleft. The postsynaptic neuron is the second neuron, which takes information from the synapse and transmits impulses away from the synapse. There are different types of synapses: axodendritic synapses, axosomatic, also axonic synapses. There's some rare types of synapses like dendrodendritic and dendrosomatic. These types of synapses are rare, but they occur between dendrite to dendrite and dendrites to soma, respectively. The names of the synapses tell you what synapses. So for an axodendronic synapse, the axon and the dendrites synapse. They are also in the order of presynaptic structure to postsynaptic structure. So for axodendritic synapses again, the axon would be the presynaptic structure that synapses with the dendrites or postsynaptic structure. Axosomatic occurs between an axon of a presynaptic neuron and the cell body of the postsynaptic neuron, whereas the axoaxonic is axon-to-axon synapses, so we'd have a presynaptic axon and a postsynaptic axon that synapse together. So types of synapses can be seen here in this image. For example, with the red axosomatic synapse, we see that the axon terminals are going to the soma or cell body of the postsynaptic neuron. There's also different types of synapses if we were to look at, say, what they're using to communicate, and it could be an electrical signal or a chemical. You are more familiar with the chemical synapses. That's because electrical synapses are much less common. They occur mainly in the central nervous system. They help with arousal from sleep, mental attention, and emotions and memory. So they're associated with the reticular activating system as well as the limbic system. But they're going to reflect exchange of chemical ions through channels, much like that we saw with depolarization and repolarization. Chemical synapses are like those that occur at the neuromuscular junction. They're specialized for release and reception of chemicals. The chemical is called a neurotransmitter, and they usually have three parts, which should sound familiar if you remember the parts of a synapse in Chapter 9. So we have the axonal terminal, also called axon terminals. That would be in the presynaptic neuron. Those are the branches of an axon that occur at the end of the axon. They're going to have neurotransmitter inside synaptic vesicles, and usually when a voltage reaches the axon terminals, calcium influx causes the exocytosis of the synaptic vesicles and release of neurotransmitter. There's a receptor region. In the neuromuscular junction, this was the junctional folds of the motor end plate. For a postsynaptic neuron, this is most likely going to be the dendrite or the soma. So the postsynaptic neuron would contain specific receptors for the specific neurotransmitter to which they are synapsing. And these two areas are always separated from one another through the synaptic cleft. So the three regions of a chemical synapse are the axon terminals, receptor region, and the space called the synaptic cleft. Terminology should be very familiar from Chapter 9. Synaptic cleft information transfer involves nerve impulses reaching the axon terminal, opening voltage-gated calcium channels, and allowing an influx of calcium. The influx of calcium into the axon terminals causes the efflux or exocytosis of the neurotransmitter in the synaptic vesicles of the axon terminals. As the neurotransmitter is released from the axon terminals, it travels across the space or synaptic cleft and then binds to the receptors on the receptor region of the postsynaptic neuron. Generally, this receptor region is in the dendrites or cell body, and once it binds, it will open a channel. It will increase the permeability of that neuron when it opens the channel, and because we need a chemical, like a neurotransmitter, to bind to these channels on the receptor region, these are ligand-gated channels. The signal can be excitatory or inhibitory. That means that when a neurotransmitter binds to ligand-gated channels, it does not always initiate a graded potential that will eventually cause an action potential. It could initiate an inhibitory signal that will stop the creation or turn off the neuron and prevent it from forming an action potential. So let's go through the steps one by one. Here in this image, we can see our major components for a synapse. Here is the end of an axon up here. One branch is being shown to you. So this is one axon terminal. The axon terminal is filled with synaptic vesicles. In the synaptic vesicles are neurotransmitter. On the sides of the axon terminal, we can see voltage-gated calcium channels. We also see a space between the axon terminal and the receptor region of this neuron. The receptor region of this neuron is on the cell body. If we take a close look at the receptor region, we see a ligand-gated channel that, when open, would allow the influx of cations. That influx of cations, like sodium, would generate a local depolarization. So in Chapter 9, it was a local end-plate potential. On a neuron, it would be a local depolarization that, if strong enough, would be a graded potential. So step 1, the action potential arrives at the axon terminal. Voltage-gated calcium channels open, calcium enters the axon terminal. The calcium entry causes synaptic vesicles to release their neurotransmitter by exocytosis. Then the neurotransmitter diffuses across the synaptic cleft and binds to its specific ligand-gated receptor on the postsynaptic membrane. To summarize that, the electrical signal reaches the axon terminal. The electrical signal opens calcium channels. When calcium channels open, calcium goes into the axon terminal. Calcium in means neurotransmitter out. The neurotransmitter leaves via exocytosis of synaptic vesicles. The neurotransmitter travels across the synaptic cleft or space and binds to its receptor. Since a chemical is binding to a receptor to open it, the receptor is a ligand-gated receptor. The receptor is located on the receptive region of the postsynaptic neuron. Here we can see the ligand-gated channel. We can see that the blue dot there, acting as the ligand or neurotransmitter, bound to the receptor and caused this receptor to open. So that means not only is this a receptor for the neurotransmitter, it's also a channel. When the channel opens, we see ion movement through it. We would see both influx and efflux of cations, but a lot more influx, and sodium influx can create or result in a graded potential. Graded potentials are short-term signals that, if strong enough, can generate an action potential, but we still have to get rid of the neurotransmitter bound to that receptor. We would not want it to be bound permanently to its receptor because then we wouldn't be able to reset this neuron and reuse it again. So different ways we can get rid of neurotransmitters are enzymatic degradation, reuptake, and sometimes they just diffuse away from the synapse. The neurotransmitter that is bound to the postsynaptic neuron will be able to produce a postsynaptic effect until we remove it. So that's why we must remove the neurotransmitter so that we don't block the reception of additional messages. So here you just have in your PowerPoint, again, that list. There is degradation by enzymes, reuptake by cells, whether it's the presynaptic neuron or astrocytes, a neuroglial cell, and diffusion away from the synaptic cleft. All three are possible, and more than one can occur in order to terminate neurotransmitter effects on a postsynaptic neuron or effector cell. Here again, let's just mention the three parts of the synapse. We can't forget the space, the synaptic cleft, or the presynaptic axon terminal or postsynaptic receptive region. Now, if we were to look at how long does it take to create an action potential and get this signal from one neuron to another, we would say there's a slight delay at the synapse. So this is going to be what we call our kind of comparatively slow step. So nerve impulses travel about 300 miles per hour. So neuronal transmission through the axon is very quick. But we see that the information jumping across that synapse or traveling from presynaptic neuron to postsynaptic neuron or effector is much more slow than the nerve impulse itself. So in order to generate a neuronal impulse, a neurotransmitter must be released, go across a space, bind to a receptor, open the receptor, let ions flow in and out. So there's a delay. There's a rate-limiting step during neuronal transmission, and it's at the synapse. So all we're talking about here with synaptic delay is that throughout neuronal transmission, it is the information crossing the synapse that takes the longest. And then one of our last topics is going to be different types of neurotransmitters. Neurotransmitters are the language of the nervous system. There's about 50 or more that have been identified, and most neurons make 2 or more neurotransmitters. They can be stimulatory or inhibitory depending on the effector or postsynaptic neuron they bind to, and we generally classify them by chemical structure and by function. So we'll start with acetylcholine. You should be slightly familiar with it from chapter 9. The reason we talk about acetylcholine a lot is it was the first identified and best understood, currently, neurotransmitter. It's released at neuromuscular junctions by somatic motor neurons. It also is released by some autonomic nervous system neurons, and sometimes even in the central nervous system, but most commonly, again, at the neuromuscular junction. It's made of acetic acid and choline, and then we would break down acetylcholine using the enzyme acetylcholinesterase. Acetylcholinesterase is an enzyme that would degrade acetylcholine into acetic acid and choline. We also have biogenic amines. The biogenic amines include catecholamines and indolamines. They include a lot of our happy neurotransmitters like dopamine, serotonin, but also histamine, norepinephrine, and epinephrine. Now, norepinephrine and epinephrine can act as hormones when released into the blood. You would think of norepinephrine and epinephrine as adrenaline when released into the blood. We find that the biogenic amines are broadly distributed in the brain, so they're most commonly found in the central nervous system. Some autonomic nervous system motor neurons can release norepinephrine, and if they release norepinephrine, they're considered adrenergic fibers. The imbalances associated with mental illness are generally because of either overactive dopamine secretion or sometimes underactive production of the indolamines or catecholamines. But for example, here, overactive dopamine production occurs and is associated with schizophrenia. There's also some amino acid neurotransmitters. These are really small neurotransmitters. They either consist of only a couple of amino acids bound together or just one amino acid, like glutamate, aspartate, and glycine, whereas gamma is a derivative of another amino acid, and GABA stands for gamma lambda aminobutyric acid. And all amino acid neurotransmitters are found only in the central nervous system. The neuropeptides include substance P, which is a mediator of pain signals, endorphins, so endorphins such as beta endorphin, which is associated with runner's high, or dynorphin and enkephalins. Enkephalins are produced during labor, and they all act as natural opiates, so they're going to reduce pain perception, hence why substance P, a mediator of pain signals, also included. You also have gut-brain peptides produced by the myenteric and submucosal plexi of the alimentary canal. Gut-brain peptides include somatostatin and CCK, or cholecystokinin. And then we have purines. An example of a purine is ATP. So it includes adenosine plus some phosphates, and it's generally an inhibitor in the brain. So we know that the reason that caffeine is stimulatory is because it blocks adenosine receptors, thereby freeing up some ATP to act in the body in order to energize or provide neuronal cells with more energy in order to carry out metabolism. We see that ATP acts both in the central nervous system and peripheral nervous system, but we also know ATP as the energy molecule for cells, so it's found all throughout the body. It can produce fast or slow responses depending on the type of neuron signaling that is received from the presynaptic neuron. It does induce calcium influx in astrocytes, so it communicates a lot with the neuroglial cells. Then we have our gasotransmitters, which are gas or lipid neurotransmitters. The most important of which to know is nitric oxide. In the blood, nitric oxide is a vasodilator. but it's commonly going to be involved in learning and formation of new memories in the brain. But we don't want to forget it's a potent vasodilator, because if it does leak from neuronal tissue into vascular tissue that we find in the central nervous system, it can lead to excess buildup in nearby tissues, and that brain damage associated with the symptoms of stroke patients. It also causes smooth muscle relaxation in the intestines, so we just want to monitor how much nitric oxide is building up near vascular structures in the brain and in peripheral tissues. Endocannabinoids act on the same receptors as the active ingredient in marijuana, most common because they are G protein linked receptors in the brain, and they act via second messenger signals or symptoms, and they're lipid soluble and synthesized on demand. We think that they're involved in learning and memory. We don't know a lot about them. We do know they're developmental in nature. They control things like appetite and suppressing nausea, but again, they're usually recognized when talked about because they bind to the same receptor as THC. Now, that was looking at the classification of neurotransmitters based on what they do, based on kind of what they look like. Are they amino acids? Do they have this functional group? But we also want to know whether they're excitatory or inhibitory. Excitatory neurotransmitters cause depolarizations. Inhibitory neurotransmitters cause hyperpolarizations. So excitatory neurotransmitters create excitatory postsynaptic potentials, meaning they change membrane potential in a way that creates a graded potential that can lead to an action potential. We would call an excitatory postsynaptic potential an EPSP. Inhibitory neurotransmitters generate inhibitory postsynaptic potentials, or they create inhibitory signals unlikely to cause action potentials. They're going to create hyperpolarizations that prevent the formation of an action potential along the axon. So an excitatory neurotransmitter causes an excitatory response in the postsynaptic neuron, therefore changing the postsynaptic potential to be an excitatory signal that can cause an action potential. Whereas inhibitory neurotransmitters are not going to cause a depolarization, they will cause a membrane change that proceeds in the opposite direction, instead causing the inside of the membrane to become more negative and a lot less likely to cause a depolarization or action potential-like signal. So inhibitory postsynaptic potentials are going to prevent action potentials from occurring or keep the membrane at a reset stage. Then some neurotransmitters can be excitatory or inhibitory, and that's because it depends on what effector they're binding to. For example, acetylcholine acting on skeletal muscle is always excitatory, but if acetylcholine bound to cardiac muscle, it would be inhibitory. So we would create an excitatory postsynaptic potential on skeletal muscle if acetylcholine was the ligand that bound to its receptors. We would create an inhibitory postsynaptic potential on cardiac muscle if it was acetylcholine that bound to its receptors. So acetylcholine will cause skeletal muscle contraction because it's excitatory for skeletal muscle, but it would cause cardiac muscle relaxation or decrease heart rate because it has an inhibitory effect on that type of muscle. We also want to talk about how neurons communicate with each other. We know it's through synapses, but we can arrange synapses in different ways. There's a couple of different patterns of synaptic connections or types of neuronal circuits. There's a divergent circuit where one incoming neuron stimulates multiple numbers of fibers, and then those postsynaptic fibers generate even more postsynaptic fibers. We see that divergent circuits are amplifying circuits. Convergent circuits are the opposite of divergent circuits, and they result in either strong stimulation or strong inhibition. We see here over on the right that we have multiple inputs in a convergent circuit going to one output. So we have multiple neurons converge on one postsynaptic neuron. Reverberating or oscillating neuronal circuits are when we have collateral synapses that generally form pathways in the brain, and then proceed to have a positive feedback mechanism that communicates back towards an earlier part in the neuronal pathway to influence it in some way. Parallel after discharge neuronal circuits are when incoming neurons stimulate several neurons along parallel pathways. So it's not just one neuronal circuit that we have, but multiple neuronal pathways that then eventually converge on a singular output. And then our last topic is one homeostatic imbalance, that is multiple sclerosis, or MS. MS is an autoimmune disease that affects primarily young adults. Myelin sheaths in the central nervous system are destroyed, meaning that the myelin sheaths that are attacked are made by oligodendrocytes. It turns the myelin sheath into hardened lesions called scleroses, hence the name multiple sclerosis for this disorder. It will slow impulse conduction, it'll cease, and even though the demyelination doesn't immediately cause visual disturbances or muscle weakness, it can cause an increase in sodium channels as these neurons try to compensate for the destruction of their myelin. But eventually, even the increase in sodium channels is not enough to account for all of the hardened lesions or scleroses that are forming, and true symptoms such as visual disturbances, making it hard to see, muscle weakness, making it hard to coordinate different actions of the body, and loss of muscular control in the bladder and even in the oral cavity or the mouth, making it hard to speak. All of those can lead to symptoms that are hard to treat even with drugs that modify the immune system's activity. So prevention includes high blood levels of vitamin D. We know that reduces the risk of development for multiple sclerosis, but unfortunately there's generally cycles of relapse and remission with multiple sclerosis, with the symptoms of muscle disturbances and inability to control muscle action just getting progressively worse. So here we can see in this image hardened lesions, the scleroses, at the bottom, and that the nerve transmission is interrupted when these hardened lesions become severe. So a normal myelinated neuron in the central nervous system would not see that signal from the brain interrupted. We do with the hardened scleroses created by multiple sclerosis. It damages the oligodendrocytes that create myelin in the central nervous system. So just be familiar with multiple sclerosis. Know that it affects the oligodendrocytes, that the oligodendrocytes are found in the central nervous system, and that it leads to degenerative muscle activity. And that is it for chapter 11. So make sure you review all 3 parts of the PowerPoint.